Okay, so today, almost the last day of this marathon, and I'm going to talk about X-ray diffraction, and in particular, single crystal diffraction, and in particular, macromolecular crystallography. My name is Heroux. I'm a beamline scientist at Elettra, and beamline XRD2. And just for quick reference, my background is in chemistry. But very early on, I decided that I was focusing on crystallography. So then I did, MX or protein crystallography at the time. That's what it was called. I just see that I have unstable network, so I'm hoping everything will be fine. So let's start. So basically, please interrupt me at any time. I would rather, not cover everything I want to say today, but answer your questions. Since this is my first time that I participate in this course, I have no real particular, idea of who I am talking to. I am assuming I'm not talking to biologists or, people already doing crystallography, but I might be wrong. So X-ray diffraction. So what I'm going to talk about is MX, so macromolecular crystallography, which is basically just, a subset of what small molecule crystallography is doing. And basically the difference is, not as much as we can think of. So the difference would be, the number of atoms that we're looking at. For MX, we don't think about atoms, we think about residues. So we already make an assumption there because, alanine, or you already have a subset of atoms included in the residues. For a small molecule, which is actually kind of big, you can have 100 atoms, but for a a protein, you can have thousands of atoms actually in there. For small molecules, the resolution that a normal good structure will be, you're talking about 0.7 angstrom resolution. But we basically never get there for protein crystallography. So if you have anything in between 4 and 1.5 angstrom resolution, that's a good structure. And if you have a 1 angstrom resolution, that's like, wow. And the big difference is that the higher resolution you will have, which correspond to the smaller number, then you will have a lot more reflection that you can refine your parameters with. So again, for MX, you will need to have a lot of assumption in place. The size of the crystals can be comparable, but one of the big difference will be the solvent content. So how much water you have included. And when you think about all the solvent water, they might not be, they will not be ordered. So you will have a lot of empty space inside your structure. So the solvent region. And basically less contact within all the other atoms at the surface. One of the things that, as chemist, and the first time I looked at a talk about, protein crystallography, I realized that for 1 hour, people just talk about hydrogen bonds and hydrogen this and this and that. And being a small molecule person, I was like, they don't even see the hydrogen. So that was also a very big difference. So all the biological, reaction will probably involve hydrogens that you need to calculate. So again, a lot of assumption into MX crystallography. So I have 39 slides, but I'm going to go fast on some of them. So please interrupt me. Where do we do the experiment? So for the MX world, most of the experiment are done at synchrotrons. And synchrotrons, I just show here on the map where those synchrotrons are located. So you have, a fair amount of them in America, North America, South America, one in Canada. You have then, on the other side of the globe, and a big concentration of them in Europe. And you can see the yellow dot is Elettra. And the red dot is one beamline that's actually in construction at Solaris to do MX. So this is, very standard. So this is just synchrotrons where you have MX. They have other synchrotrons where, they don't have a focus on this. And at each synchrotron, you probably have more than one beamline dedicated at doing MX. And this is just, talking about a very large community of scientists that, is using this technique to get answers. And we can focus now on what exactly do we want to do. So MX is going to be done, as it says, on macromolecular. So not just protein as before, but you're going to have DNA, RNA, and you're going to have complex. And, the biologist or enzymologist or people working in this field, they have biological question. And even though, we all know about the small little crystals, this is actually a huge investment that people are doing to make crystals. So whatever technique you're going to use to actually get answers to this biological question, it will depend a lot on what the state of your sample is. And for doing MX, you need single crystal. And this is, very, still very unclear how to actually proceed and get good crystals. They can look really good and they can diffract really bad. So you just never know. You need to go to the synchrotron and you need to test them. So this is a big process, a huge investment. If you think about diamond crystals, they are really cheap compared to the cost of making these crystals. And, you have a wide range of reasons, covering all your question, enzymatic pathway, you need to have drug designs. I mean, there's been a huge push on getting structure for SARS-CoV over the last year. So a lot of the beamlines have dedicated a huge amount of time to doing those structures. And just yesterday I was listening, and at Diamond Light Source, they collected over a year more than 14,000 data sets. And for that, they have, less than 5% hits of something inside that they actually, crystallize with. So there's a lot of structures with nothing that you want to see. So you want to go from crystal to a model. And unlike other, discipline where you have a priceless, artifact, or you have a sample that is already there and you want to study, in the MX world, you need to create your sample from nothing, from a sequence, basically. And, the steps are very long and very, time consuming, but energy consuming before getting to the synchrotron. And the synchrotron part is actually a very short time in your experiment. So most of the people are used to see, those structures. They're basically standard, this ribbon representation of a protein at this point. And, but in reality, what you get is more something like this, which is not necessarily very obvious for people that don't stare at that all day long. So this is an electron density map. And basically what we are going to do to get this ribbon diagram of the protein is we're going to have to build our model inside this electron density. And depending on the quality of this map, then we can actually build more accurately or more complete, So we will come back to the map a little bit later. So again, we're starting here with the single X-ray diffraction with what are the experiment, and the bottleneck is the crystal. Again, you can do this, a lot of the experiment in a home source because they're really, nice and powerful. But at this point, they really cost a lot of money for facilities, and there's less and less of them being used at university. So we get more kind of screening at the beamline because it goes fast and we are very well equipped for this. So we all see in publication those very nice crystals. I don't know if you can see the nice crystals in mother liquor. So all our samples here are going to be in a solution, and we need to freeze those solution. To be clear, we don't want any ice, as you can see at the bottom. There's another crystal there. It's sticking out a little bit in a mass of ice because they will survive longer in the beam. These crystals have a lot of water, and then you just blast them with the intense beam. So basically, you are going to destroy your sample. Having crystal is a first step, but very often they're not single. As you can see on the picture here, you can definitely see cracks. You can, they're not pretty, but a lot of the time we can still work with this. They're not perfect, but as long as the diffraction pattern are separate, we can work with this. So having crystals is one step, and this is something that we don't exactly know. We have screened to get those crystals. We will vary the pH. You need to vary the concentration of protein, concentration of salt. So this is a trial and error, and you have to optimize these steps, and this can take a fair amount of time, several trips to the synchrotron. So this is the starting point. We have crystal. Then we come at the beamline, and in this presentation, if I'm talking a little bit more about the technical aspect of it. So most of the facilities have very, very standard equipment because we have the same vendors and everything, and it's also extremely easy for one user to go from one facility to the other, and it's always a question of getting beam time and making sure that your project gets some answers and move along. So most of the facilities have robot to mount your samples, and we have this because a data collection is very short, and we need, normally users send crystals frozen, so they will send up to 142 crystals per beam time session, and these beam time session can be pretty short. So we have the standard setup, but we also have standard software. So a lot of the facilities have agreed to use the same software for the data collection, and you can see down there, this is MX-Cube. You can find it at ESRF, you can find Soleil, Alba, and a lot of other facilities. So the users can arrive and start data collection immediately. They feel comfortable, they know what to do. This is also a software that you can use remotely. So this is in the web browser. You will just type the IP address that we give you, and you can collect remotely. This is standard now at most of the facilities that you do, and that has been a standard for almost 10 years now. So this is not new, but with the pandemic, that's been a reality that we need to do. So you have your loop here, which will have your crystal in, and then this is where you're going to have the sample is mounted. This is going to be with, you can see the nozzle of the cryostream. So it's going to be 100K for the whole data collection. And at Elettra, the full cycle of mount, center sample, rotate, expose, unmount will take usually about 10 minutes. 6 minutes of that is the full data collection for a whole sphere. We are not a facility that runs very fast, which this is very fast, but not the new facilities at Diamond or ESRF. The crystal data collection will be less than a minute. So and it basically is dead by that time. So we went through last 15 years to, a data collection that would take 3, to 12 hours to a data collection that takes only 1 minute. We went from being able to collect a full data set by exposing, 20, 40 crystal to now being able to collect everything in 1 minute. So the field is mature, but this is because of incremental, improvement in how we deal. We see a problem, so crystals are not surviving. Okay, now we cryocool them, they survive longer. We have a problem of the data collection being 12-hour long. That means that the synchrotron radiation was decaying during that time. Now synchrotrons are very stable with top-up mode, so with the intensities, well, the rotation will be the same. So it's mature, but it's evolving very rapidly, and the users need to adapt to the new ways of doing things and be able to keep up. So if you have 142 data set within 2 or 3 hours, now the problem is to be able for a human to manage all of that data and be able to organize. So there's database now in place that will help you manage your experiments and figure out the best results that you have over those. Okay, so now let's get back. So I talked about the data collection. So we rotate the sample, usually 1 degree, 0.5 degree, a little bit, and we expose. And this is what we get for the diffraction pattern. So you will see, different diffraction pattern, when Jasper after me will talk. But in our case, if we just look at this and, we saw, you heard Heinz when, we're talking about the socks, and he presented the Bragg law. And obviously, for me, but you can see that, the spacing that you're looking at. And it's basically the X-rays going through your sample. Most of it will just go through, will hit nothing. So, we block the direct beam, which would be absolutely, you're talking about 99% of the beam going through. And the 1% that will be diffracting is what you are actually seeing here in the pattern. If you're not a crystallographer, and this for me looks good, I know immediately that I have a good crystals. You can see that the diffraction is going, the higher resolution will be, the higher angle, basically, that you have spots. And you can see that it's, you have a very clear pattern. You have looms, you have, a line spot. This is not the real spot. I put, yellow circles so you can actually see better. But, you can see the shape of the spots, and it will give you information about, the quality of the packing in your crystal, the quality of your crystal. So, once you have, this is only one diffraction pattern, and you're going to be rotating and get another one. So basically, you're going to accumulate over the whole sphere, all, the diffraction spots. And those spots, are going to be assigned an address, basically Miller index, the HKL of the spots. And if we look at that a little bit clearer, you can see that some of them are in a line, and zoom in, we're on a line and everything. So every one of them will have an HKL indices. So from this, we need to, and we will get 2 different information. And the two of them are very important. So the position is kind of easy to get. So we can measure very accurately the position of them. And this will actually give us information about the unit cell that we have. And how big and the ABC and the angles in between. So these spots, that you have, correspond to an oscillation, a zero, a rotation, basically degree oscillation. And it can be in between. Maybe the spot is spread over a wider angle and everything. So we will need to be able to get the accurate position. And then we are going to integrate the intensity of each of those spots to be able to get the position of the atoms that are in the cell that we had. So this step, is done at the beamline. So this is, you need to be, integrating some of your data. So, which is processing the data, will be indexing, giving spot, finding the cell parameters for your data set, then the integration of all of these intensities. And, depending, it's absolutely normal to have an isotropic diffraction. It will diffract very well in one orientation. And when you get 90 degrees away, the diffraction limit just shrunk and then go back up. So we needed to, scale this intensity and merge the data. So this is the first step in a chain, in a pipeline of software that we need to use to get to the structure. So I wanted to talk a little bit about the HCal indices because, this used to be extremely important before. When a data set was taking 12 hours, we needed to know that we had the minimum, to get a full, data set. This is not the case anymore. And we don't need to run a strategy to saying, oh, we need to start at this angle. And if you start at 12 and go all the way to 92, you get all the minimum, all the unique reflection. Nowadays, within a couple of minutes, you can collect the whole sphere. But it's still ingrained in crystallographers to talk about strategy and space group and where to start. At this point, it goes very fast. So again, we have built up a sphere, reciprocal space of dots and everything. And if you think about the symmetry that you have, and we're not going to go in there, but you have indices that are equivalent. And, it's just the symmetry that you have inside makes it that you can, if you've measured that one and that one, basically you will merge and you will get better signal to noise for this intensity. This is crucial to get, the position of the atoms that you want to look at. So it used to be very important to do this, but nowadays it has evolved and it's not as much. The important part is to, be able to make your crystal last long enough to get enough data to be complete, but also redundant, and you will get, better maps at the end. So we are talking about, the intensity, and the intensity will basically depend on how heavy, how many electrons you have in your atoms present. And for macromolecular crystallography, unfortunately, we only have very light atoms. So, you have nitrogen, carbon, oxygen, hydrogen, and very often you might have, zinc or something a little bit, some sulfur, very few of them. But, we don't get very different scattering factor for the atoms that are all in there. And there's a lot of them. So, finding, being able to figure out the intensity and most importantly, the structure factor, because every atoms that are in your protein will contribute to the intensity of the spots. Sometimes very little, but in some orientation of your crystal, when go through and you hit on some of particular position, then you will have a lot more contribution from certain atoms. And as Heinz also mentioned, we have what we call the phase problem. So we know we can measure the intensity, but it's a lot more complicated to figure out the structure factor, the contribution of the scattering of each atom, for that spot. So this is the problem that we have to deal with. Very often you will hear crystallographers, oh yeah, I solved my structure. And the idea is, what does that mean exactly? You solved your structure. Solving a structure is basically getting, your first electron density map. If you have no information about your protein, you just get a starting point. And we know that, the spot's intensity will be related to the position of the atoms. And basically the contribution, we have mapped this in space. So if we, kind of go back, we can have, in the particular, cell that you can have an electron density calculated over, all of the space. So how do we get there? And there's several methods. Over time, some of them were more favored than others. And basically, you can have, the direct methods. This is used for small molecules. And, it's because they have a lot of reflection, and they can, get very fine details. We have molecular replacement, heavy atom isomorphous replacement. I mean, you have a whole, choice of doing this. And some of them, require that you modify your crystal. So you start back at the beginning. Now you have diffraction, you can do, different things. So how do we get the map? In practice, at the synchrotron, there's two things that are the favorite method, to do it. Not that the other methods are bad. It's just some of them require a lot more, wet lab. Some of them are extremely difficult to handle crystallography speaking. And, at this point, these method works great. So I'm going to focus a little bit on molecular replacement and SAD data collection. And one is not better than the other, and one is not necessarily more simple than the other. Actually, the SAD one is, in my opinion, a lot simpler than molecular replacement, but it required that you have, selenomethionine in your protein. So molecular replacement. Molecular replacement means that you can just have the crystals from, your protein, and that you think that you have a good starting model. So something that is homologous from a different organism, the same function, but you already know the sequence. So it's a little bit different. It's 90% the same. I mean, so you think you have a good model and you hope that it has the same conformation that the protein that you have. So it's basically, you already have positions for your, protein, your monomer that you have, or dimer, depending on what kind of, assembly it's making. And you're going to make some vectors. And it's a little bit a brute force approach, because you're going to be rotating this and says, oh, this is already a good, orientation. And then you're going to go and translate that into your cell. Of course, you're going to have some criteria, like, molecules should not be bumping into each other. And, so there's, several different programs that you can, use to get an answer. And, and it's basically, you're going to be able to fill, your cell with the symmetry molecules and calculate and see, does these intensity actually correspond somewhat to what we are doing. Of course, this is a little bit more complicated because proteins are extremely flexible. So even though it's the same protein with, one different mutant, it will breathe, it will have a different conformation. And very often this is what you actually want to study. So we also have a big bias, is that if you already impose a model, you are actually going to see that model in your map. So you have to be careful. You have to remove some of the side chains. And if they appear back in the map, then you know it's real. But the more atoms you remove from your model, then the least accounted atoms you have to create that map. So it's not as easy as one can think. So, and this is just an example over here of, a protein that I worked on. And you have a monomer here, and it has 230 residue. And you can see all the flexible loops that you have here. And this part here, the hood, is actually opening and closing, and it can rotate a little bit. And it accounts for about, a third of the atoms in there. So if I impose a model that's already wrong, it will be very difficult for me to figure out if the answer I get at the end is right. And you can see that this protein with one mutant, so one residue out of 230 would give, different solution, different crystal in different space group. And even when it's in the same space group, it would give different cell parameters. So every single time I had to solve the structure. But on the other hand, if you have crystals and you soak them and you do something and you get the same cell parameters, same space group, then you don't need to solve. It's already solved. You can move on and make different maps. So the second method we use at the synchrotron very often is selenomethionine. And you probably have heard all about the SAD data set. So again, mentioned before, we have usually only very light atoms. And if you replace one of the sulfur with a selenium atom, suddenly you have much bigger atoms. And at this point, you can really use and exploit the great thing about you being at the synchrotron is that you can tune the wavelength that you're going to be collecting. So here you have, the scattering curve that you can see with whatever energy you're using. And, you can see immediately if you're talking to a crystallographer or a physicist, because physicists will talk about energy and crystallographer will talk about wavelength. So over here, we can see that for, the behavior with the wavelength changes. And those are going to be very small changes. And it will actually make that the reflection that are supposed to be equivalent are not quite equivalent. And at this point, we have detectors that have signal to noise very good, and we can measure this reflection, with enough accuracy that you can pick up all of the small differences that comes from anomalous dispersion over here. The strategy that you're going to use is going to be different at this point because you want to really capture very small difference in the intensity. Then redundancy is your friend. You want to be able to measure those reflection and, with, all of the merging and everything, you can really get very, accurate differences in between your Friedel pairs. So the tendency of crystallographer is to always want higher resolution. So you always want to push it a tiny bit, give it a tiny bit more exposure time, and your crystal will decay. And normally the decay occurs at exactly the interesting residue that you're looking at. So collecting very gently a moderate resolution map will, as a starting point, and once you know that you have this starting point, then you can move on with your next crystals and actually really lean on them and get the high resolution that you want. So, the data collection are very similar, but there's subtle things to change. We need to change the distance on the detector, separate those spots, reduce, the signal to noise, tune the wavelength to the right position. This is, selenomethionine. So if you have selenium, you want to be collecting, and people tend to say, I want to collect right here at the peak. But in reality, if you do a scan, it will look like this. So collecting just a little bit off will, basically just change everything. So it's safer to go a little bit away from the peak and basically get your anomalous signal at that point, because this signal will depend on the environment of your selenium and, for the data collection. You have to keep in mind that you might have other things that have anomalous signal in your crystal. So very often people do selenomethionine and they have zinc in there, or they want to do phosphor or a sad sulfur, and which is a puny signal, but they have a huge zinc signal, but they were not able to phase with zinc, or they didn't think to try to phase with zinc. So we look at those two methods of basically getting, a starting point. And the starting point is this. This is the map that you're going to have to deal with. And this is, at this point, you're probably already at the lab. You have collected. If you were good enough at the beamline and, more people to help, you might have this map already done at the beamline. How do you know if your structure is solved? So there's, like for the diffraction pattern, you have a pattern, and when you recognize it, you can see if you have good very clear, very quickly. For a map, it's the same thing. It's not random. If you have, density a little bit everywhere, it didn't work, because you should have clear solvent pockets, a lot of solvent where you see almost no, electron density. And you can see over here that you have your protein. And even better, if you start building, and there's programs that will do that automatically for you. They just connect and they build, the C alpha in there. There is absolutely nothing random about building a helix. If you see helixes, you're good. You have solved, you have a starting point. And, and the density might be good enough to, build a lot, or just a starting point, So once you have your map, and the quality of your map will make a big difference in how fast you can build in it. So here we have a 1 angstrom resolution map. And to us, it looks a little bit funny, because you're like, oh no, I have disconnected density. Well, that's because you see the atoms. You can position very accurately where the atoms are. So there's usually no ambiguity where you need to put and build your atoms. If we look at 1.5, which is also a great map, and every crystallographer will like, ooh, there's a hole in the, this, you can see actually, a hole into any aromatic component. That is awesome. But the real thing is when you have a map that's not that great. So here we have 2.3, which is, in my opinion, where, there's a clear good, and the rest is like, challenging density. With 2.3, you can have carbonyl bumps. And basically, this is, you can still get a lot of information, and the information is going to be accurate about the bond length and everything. But, you're going to have good electron density regions and not so good. Wherever there's anything flexible, then the electron density is not going to be that great. But this is going to make the building phase of your model a lot easier if you have higher resolution. So you have your starting point. Now we're going to go through the refinement stage, and it might not be as exciting as you think. So this will include making maps and building an initial model. From that initial model, you can recalculate with this position what structure factor it corresponds to. And now you need to, to just basically make a difference in between what you have observed and what you are calculating. And like any refinement, you want to make those two as close as possible. So there will be several rounds of, building first your main chain, then adding all the side chains, and then rotating all the rotamers for your side chains, and then, adding some of the water molecules you see near the active site, and then outside, and so forth. So there's a very clear path, but the question is, where do we stop? When is good enough? When you are starting to be overfitting a little bit your data? There's plenty of hints into the log file that you will see, and this is where being a crystallographer will help you a lot to know where to stop. So now you have a model, and one model is rarely enough. You have a snapshot in time of your protein with nothing in APO, the hollow enzyme. You can have products, you can need to have intermediate, you can need a picture of what the product is, or just interaction with different subunit, different partners that you have. So one structure is rarely enough to actually get a full story that can be published. But one thing for sure, when you are looking at a publication, you will get table 1. And in table 1, this is where you're going to get all the information that you need to make sure that the model they have is actually relevant to what they're trying to address the issue. And it is a tiny bit obsolete at this point, because all you really need is the PDB code access. MX is very mature, so all of our data, all of the models to be published need to be deposited in the PDB databank. And even if you don't do crystallography at a synchrotron, being able to navigate that database, you would get so much information. You can download the coordinates, and within a couple minutes you can have the software to actually move and regenerate the maps, so that you can look for yourself and figure out already a lot of information about the system that you want to look at, or really assess the quality of what the people are talking about in the publication. If it's irrelevant, if it's relevant to your field, take the time. You have a lot of tools available there. So that table 1, gives you the, PDB code, space groups, and all kinds of information about the quality of diffraction, unit cells, and all the reflection. But also, the overall quality of the end model, how many proteins, and so forth. So this is a standard, and if you get a little bit familiar, with this, you can very quickly figure out what is the quality of the data. So, in summary, I'm going to stop here. What macromolecular crystallography is done at synchrotron, and it's done because you can tune the wavelength. Most of the data set don't need to tune the wavelength. You can basically collect at whatever wavelength you are at, but you can exploit, that part if you're going to do a SAD data set. You will need to go through a lot of samples very quickly, and you need to make decision about what to do with that sample. And it's subtle decision. You exposed, 0.5 seconds more per degree, or you back up your detector, or you change the wavelength. Those decisions you will have to make them every 10 minutes, because this is how long it takes to move from one sample to the other. So we have lots of software available, and sometimes it can be overwhelming because there's a lot of choices to make and decision. One of the big limitations is to have a single crystal. And, you know, if you're not a crystallographer, and we see a lot of non-crystallographer coming to collect the data, as long as you know, interact and collaborate with a crystallographer, I think, you know, there's no problem. And you can, you know, basically complement any of the studies that you are doing. You can get beam time. MX community is a little bit different than the other communities, and we need quick access because your crystals are fresh. You need to collect now on them, test them now, because you need to figure out if you need to redo all your crystallization preparation. So we do very often, but not too long. But even not too long is a huge amount of data. So, you know, there's a turnaround. Every facility have, you know, a proposal fairly specific for MX. If you don't have single crystal, you can still get a lot of information by using synchrotron, you know, other techniques that will give you either complementary, you know, information or some very clear indication of why you cannot get single crystals. If your samples, you know, aggregate, if your samples, you know, make, you know, tiny crystals and you have powder instead, if you, you know, need to do some spectroscopy, you need to, you know, modify the starting point. But all of these techniques don't exclude the other one. At one point, you can get all the information you need from all different field and beamlines that you use at the synchrotron. Beamline scientists are there for you, and we like to interact with people. And if you're not sure what you want to do, how to do it, please contact the beamline scientists. I think that most of them will be happy to help you and figure out what is the best result you can get at the beamline. And if not, we are going to point you to a different beamline or a different technique that you can use and talk to different people. This is it. I actually talked more than I wanted. If you have any question, please let me know.